Metabolism involves the oxidation of biological molecules in order to get energy. When we talk about oxidation, well, the most oxidized you can get in terms of your carbon is CO2. So, in the process of oxidation, we do a lot of decarboxylation, that is, loss of CO2. Some of the time we do this with using CoA, and some of the times we don't use CoA. So some of the times we have to use this whole big complex typified by your pyruvate dehydrogenase complex, but similar complexes are at play for other dehydrogenase complexes, such as in the citric acid cycle, your alpha-ketoglutarate dehydrogenase complex, as well as when we look at like branched chain amino acid breakdowns. All of these are going to use these complicated methods, but then we have steps such as when we are doing this isocitrate dehydrogenase step where we don't use CoA. Why? Well, it comes down to whether or not we're decarboxylating an alpha or beta keto acid. For reasons I'll get into in a second, decarboxylation of a beta keto acid is energetically favorable, whereas that of an alpha keto acid is not. So if you want to decarboxylate a beta keto acid, well, there it's no big deal. You don't need to add the CoA. But if you do want to decarboxylate an alpha keto acid, that's where you have to go through the hoops of having that big old complex with all that complexity. So what do I mean by an alpha versus a beta keto acid? It refers to the position of the ketone in relation to the carboxylic acid that you're talking about. So for example, if we talk about the central CO2, here we can see that we have a ketone that is 2 carbons away from this carboxylic acid. So we have carboxylic acid, and then the ketone is alpha beta to the carboxylic acid. So the one next to it would be the alpha position, and the one 2 away would be the beta position. So when you have a beta keto acid, this means that you have a carbon in between the carboxylic acid and the ketone. In contrast, if you have an alpha keto acid, here your ketone is right next to the carboxylic acid, so they're on neighboring carbons. Alpha keto acid, the nomenclature is just like when we talked about alpha amino acids. In an alpha amino acid, we basically have the amino group and the carboxylic acid alpha to one another. So the amino group is on the carbon that is next to the carboxylic acid. If we were to swap that amino group for a ketone, well, now we would get an alpha keto acid. And in fact, this is what we see when we do transamination reactions. So the same nomenclature applies. If something is directly next to something, it's in the alpha position. So this ketone is alpha to this carboxylic acid. But if it's 2 away, then it's beta. So this carboxylic acid, this ketone is beta to that carboxylic acid, alpha beta. This one would be alpha, but we're going to lose the beta one first, not the alpha one, because the loss of the beta one is energetically favorable, but that of the alpha one is not. Now let's look at what drives this energetic favorability. It has to do with enol-keto tautomerization. That might sound familiar to you from when we talked about glycolysis. In glycolysis, we had a pyruvate kinase step where we went from phosphoenolpyruvate to pyruvate. This step was energetically favorable because of this enol-keto tautomerization. So remember that an enol is when you have a double bond. So you have like an alkene, so double bond between 2 carbons next to an alcohol. So an enol. And then a ketone is where you have a carbonyl, so you have a C double bond O with carbons on either side of that carbon. What you can get is tautomerization, where basically you're able to go between these 2 forms. If you think about which would rather have the electrons, would it be the oxygen or the carbon? It would be the oxygen. So the keto form is going to be much more energetically favorable. And if you can go from the enol to the keto form, that's therefore going to drive a reaction. In the case of our phosphoenolpyruvate, here the phosphate group is trapping it in this enol form, or its enol equivalent form. So if you lose that phosphorus group, well, now you would have an enol that would be driven to form the keto, and that's why we can basically transfer this phosphoryl group onto ADP to make ATP and have this be energetically favorable. If we look at our carboxylic acid cycle, if you just look at the names, it's not going to be obvious why you would have some of them need CoA and some of them not, because you can't see whether things are alpha or beta keto acids. And in fact, it turns out that none of the kind of main things of the citric acid cycle are going to be beta keto acids. But if we look at an intermediate, we do see that we do have a beta keto acid. And it corresponds to the place where we're using CO2, but we're not using CoA. I'm talking about this isocitrate dehydrogenase step. The kind of like hidden intermediate is this oxalosuccinate. So it's shown in brackets because it doesn't leave the enzyme's active site, but it is not like a transition state. It's an actual intermediate. It's just so unstable that it's not actually going to leave the enzyme. And the reason why it's so like unstable is because of that. It has a beta keto acid that is kind of optimized in the enzyme's active site so that decarboxylation is going to happen very readily. So how does this intermediate form? If we look at the structure of isocitrate and we look at kind of like our end goal, this alpha-ketoglutarate, what we want to do is we want to lose this CO2. So if we look at it from a little bit of a different perspective, we want to lose this central CO2, which as we saw before was going to be a beta keto acid. This would be the alpha position. This is the beta position. Our ketone is beta to that carboxylic acid. Note that this would be an alpha keto acid, this one at the bottom, but we're not going to lose that one. Instead, we're going to lose the middle one because it's the beta keto acid. And in fact, the point of the whole like aconitase step was to set things up so that we would be able to form this beta keto acid on the bottom, on the middle carbon. So here, if we were to basically oxidize the citrate as is, well, here we would be losing the bottom CO2 rather than losing the middle CO2. And it turns out that it would be harder to actually oxidize this, this OH here than if we were to oxidize it here because of the type of alcohol it is, which I'm not going to get into here. But bottom line is this kind of sets the scene so that we can lose this CO2 as, and basically by making this a beta keto acid. The way that we're going to make it a beta keto acid is through our isocitrate dehydrogenase. The enzyme is going to help make this happen, but the molecule itself is going to be doing all the actual reactions. So what's going to happen is to get to oxalosuccinate, there you need to do the oxidation step, but not the decarboxylation step. The oxidation sets the scene for the decarboxylation. What you can see is that basically, if you oxidize this, what you form is you're forming the beta keto acid. When you have this beta keto acid, now you can think about what would happen if the CO2 were to leave. If the CO2 were to leave, now what would happen is that the electrons from this carbon-carbon bond could flow back into this carbon-carbon bond. That would form an enolate. So an enol was when we had an alkene double bond next to an alcohol. If you imagine deprotonating that, that would be an enolate. And that was what you would get initially before thinking about any sort of hydrogen transfers. In this case, this is going to then be stable. This would be stabilized by a metal in the enzyme's active site, so that this would more readily happen. What could then happen is when you have this enolate, then you could have the electron, this kind of take a proton, and then the electrons from this enolate could flow back to form the ketone. Now you would have the ketone. And so you would be in the keto form versus the enol form. And remember, we said that the keto form was going to be energetically favorable compared to the enol form or the enolate form. And so this is going to then be an energetically favorable reaction. This is going to make this reaction, drive this reaction forward. So you have the oxidation that was setting the scene by making this a beta keto acid. We have this ketone that is alpha beta to the carboxylic acid. When that CO2 leaves, the electrons from the bond to that carbon flow back in to make a carbon-carbon double bond, pushing the electrons onto the oxygen in order to make an enolate. The double bond can now pick up a proton. The electrons from the oxygen can flow back down. You reform your keto form, and voila, you've got alpha-ketoglutarate. If we look at alpha-ketoglutarate, well, now we can see that this bottom carbonyl, this bottom carboxylic acid, it's still just like it was before, it's going to be an alpha-keto acid. It was an alpha-keto acid in our oxaloacetonate, and now it's an alpha-keto acid in our alpha-ketoglutarate. Because all we did here was we lost the CO2, and then we just kind of picked up a proton, and we just kind of rearranged things a little bit. But we end up still back with an alpha-keto acid. So if we want to decarboxylate that, well, now we're going to have to go through those tricks. These tricks are best exemplified by the pyruvate dehydrogenase complex. So with the pyruvate dehydrogenase step, there too, we actually had to use the CoA. Because with pyruvate, we can see that we have an alpha-keto acid. We have a ketone that is alpha to the carboxylic acid group. To decarboxylate it, we're going to use not only the oxidations and not only the NAD+, but we're also going to use CoA. Similarly, we're going to use CoA when we have our alpha-ketoglutarate dehydrogenase complex, because here too, we're wanting to lose the alpha carboxylic acid. This complex works very complex. I'm not going to go into the details, but it involves multiple cofactors. One of them is going to be the CoA, which, remember, although it just seems like it seems just like a small little thing, it's actually pretty big and bulky, and so it can't actually pass through the mitochondrial matrix membrane, the intermitochondrial membrane. And so that's why we have to do conversions, like swaps to CoA inside the mitochondria or outside the mitochondria, but we can't take something that's attached to CoA, like acetyl-CoA, into the mitochondrial matrix, which is why that pyruvate dehydrogenase step has to actually happen in our mitochondria. So we bring the pyruvate in rather than bringing the acetyl-CoA in. But the acetyl-CoA has the sulfur that's going to allow you to form these kind of like higher energy swappable linkages. Similarly, you're going to see sulfur linkages in lipoic acid, which is another one of these cofactors that's used by these dehydrogenase enzymes, the ones that are like the complex ones. And so they're actually going to have these lipoic acid attached to lysine residues, and you're going to see a series of pass-offs. But the first pass-off actually happens to an even different cofactor, the thiamine pyrophosphate, or TPP, which is the active form of vitamin B1. If you heard of like thiamine deficiency and stuff like that, this can affect the citric acid cycle as well as your pyruvate dehydrogenase complex, because those enzymes rely on this thiamine pyrophosphate. I'll post a link where you can learn more about the mechanism of this enzyme. But for now, just know that it involves a series of pass-offs, and it's complicated. And you have to go through all these hoops and tricks rather than just have a simple oxidative decarboxylation. The nice thing, though, is that this complex has a similar structure and actually interchangeable subunits depending on what you want to decarboxylate. So it has like this E1, this E2, and this E3 subunits. All this complicated gymnastics, but the same sorts of complicated gymnastics are happening when you want to decarboxylate all sorts of alpha-keto acids, such as not just our pyruvate, but our alpha-ketoglutarate and various branched chain amino acids with this like branched chain alpha-keto acid dehydrogenase complex. Basically, all of these have this like E1, E2, E3 setup. And the E1 and E2, these are going to be different depending on what it is that you're dehydrogenating. But this E3 is actually going to be shared between all of these because, well, if we look at what E3 is doing, here it's just taking the electrons and kind of like regenerating our E2, passing the electrons onto FAD to make FADH2, which then gets passed on to NAD+ to make our NADH. Because basically, no matter what sorts of things you are kind of using, at the end of the day, you get on E2 that you have this reduced lipolysine, basically that lipoic acid. You have it in a form that needs to be regenerated. It needs to be oxidized. It gets oxidized by reducing FAD to give you FADH2. In order to get the FAD back, now you've got to actually use the NAD plus to make your NADH. And because the FAD, that's like embedded in the protein, that's why we kind of need to have this pass off then to NAD plus, because if we just kept it on FADH2, this whole thing would have to kind of like, the electrons couldn't go anywhere because we couldn't regenerate this FAD H2 couldn't just like run off. It's stuck in that protein, but the NAD plus, the NADH, this can go and run off, regenerating this thing. And you need this E3 no matter what you have in your as your E1 and E2, no matter what you have as your substrate. So this E3 is really important. And you can imagine that if you have a mutation in E3, not only would that affect the breakdown of, say, your pyruvate, but also the breakdown of amino acids, and also the breakdown of your like alpha-ketoglutarate. So that would have more of an impact than if you say you just had a mutation in one of the E1 or E2 subunits. Maybe you have a problem breaking down your branched chain amino acids, but you wouldn't have a problem in breaking down your pyruvate. So the E3, those are typically going to be the more severe metabolic disorders, but any of those things, any problems with any of these complexes, any of these subunits could have a more like isolated effect specific to a particular molecule. I said, pretty complex, but if you just want to decarboxylate a beta-keto acid, well, in that case, it's going to be a lot more simple because you have that energetic favorability that comes thanks to that enol-keto tautomerization. Remember that tautomerization is kind of hidden if you don't actually see the structures up all the intermediates, including the temporary ones, including the such as the oxalosuccinate. That oxalosuccinate is you set the scene up for that, having that oxalosuccinate, for having that beta-keto acid, thanks to the aconitase step, where basically you shift the position of this OH, that in citrate was in the middle, to being on the bottom in our form of isocitrate, so that you have an alcohol that is in a position that's going to be easier to oxidize. When you oxidize it, you form that ketone. When you form the ketone, that is beta to the carboxylic acid, you've got a beta-keto acid. It's energetically favorable to decarboxylate it. You're not going to need the CoA. But to get rid of the remaining alpha-keto acid, there you are going to need the CoA. So isocitrate dehydrogenase doesn't need CoA, but alpha-ketoglutarate dehydrogenase complex and pyruvate dehydrogenase complex, they do. And so sometimes you'll see those referred to with complex in the name, sometimes not, but those are going to be the more complex ones than our simple dehydrogenase.